Hi friends, welcome to Think 360. Today we are going to discuss about DNA, mRNA and retrovirus. Please comment on this video and like, share on your Facebook and Twitter. Don't forget subscribe Think 360. Okay, DNA. In previous video, I explained about DNA structure and replication of DNA. Uh, I'm separating DNA for our convenience. This is the DNA strands. I separated DNA strands. It starts from 3 dash, 5 dash, and 5 dash, 3 dash. Okay, this is the two helicals of the DNA. During the time of transcription, 5 dash, 3 dash helical of DNA synthesized to mRNA. This is mRNA. Okay, during the transcription stage, DNA synthesize mRNA. Nucleotides of 5 3 transfer to mRNA except thiamine. All of you know, in previous video I explained about this thing: adenine, guanine, cytosine, thiamine. Okay, I explained about this adenine, guanine, cytosine and thiamine in my previous video. What is the difference? What is the basic difference of DNA and mRNA? Means in the mRNA, instead of thiamine place instead of thiamine place, uracil is present. That is the main difference between mRNA and DNA. mRNA is synthesized from 5 3 helical of DNA. Three nucleotides. Okay, three nucleotides of DNA is known as triplet codon. Okay, three nucleotides of DNA is called triplet codon. As usual, three nucleotides of mRNA is called codon. Three nucleotides of tRNA is called anticodon. Remember, these three is very important. Three nucleotides of DNA is called triplet codon. Three nucleotides of mRNA is called codon. Three nucleotides of tRNA is called anticodon. Total number of codons are the total number of codons are 64. Okay, don't confuse. Codon is called like just like AUG, UUU. This is codon. Okay, total number of codons are 64. These 64 is combinedly known as gene dictionary. These 64 total number of codons is called gene dictionary. These are combinedly known as gene dictionary. Was started by Gamow. And completed by H.G. Corna. H.G. Corane. Started by Gamow and completed by H.G. Corane. Within the cell, type of polymerase are 3 to 3. Two of them mRNA, tRNA, synthesized from DNA. Okay, these all mRNA and tRNA is synthesized by DNA. The total number of codons are 64. The total number of amino acids are 20. These are participate into formation of polypeptide chains. These 20 amino acids participate into polypeptide chains. The 60 codons were means Each amino acid is coded with 3 codons. So 20 into 3, 60, and one extra is present. I will explain next video. 61 total. Okay, these are participate into formation of polypeptide chain. Actually, amino acids is total is 22. The protein synthesis 20 is participating protein synthesis. The 60 codons were recognized 20 amino acids. So the recognized by one codon amino acid is amino acid is recognized by more than two codons. Each amino acid is recognized by more than two codons. Just like UAA, UGA, UAG, like this. Okay, and another thing, these three main very important. These three are nonsense codons. Okay, in this these three codons amino acid every amino acid codes with three codons, but these three doesn't show any reaction with amino acids. This is called nonsense codons. Nonsense codons. A tRNA, in tRNA, 20 amino acids. Each amino acid is coded with three codons. Na? AUG. This is very important codon. AUG is methionine. Okay, methionine. This is the starting codon. GUG. Okay, this is also starting codon. I will explain the differences between AUG and GUG. First of all, we will take about the AUG. In protein synthesis, AUG means methionine. Methionine, it is a starting codon. Actually, after that, this is the starting codon, and in these, any one is the ending codon. We gave the name of nonsense codon. Later on, after many days, after recognizing the importance of these nonsense codons, they changed their name as terminal codons. Okay, after that they changed the name of terminal codons or terminative codons. Okay, either of the codon of nonsense is placed on the polypeptide chain. When the protein synthesis is going on, then these three codons, any one of the codon participate in the polypeptide chain, then only the preparation of protein is complete. If it doesn't participate in the protein synthesis, the protein synthesis process will be continue like that only. When it comes, the protein synthesis action will be end, then only the polypeptide chain also end. That's what, after that they recognize the importance of these codons and they gave the name of terminative codons. It is the one of the main important thing about the AUG methionine. And next, I already told you GUG also starting codon. Where it is starting codon? It is starting codon in prokaryotes. Prokaryotes like bacteria, blue-green algae, mitochondria, chloroplast. In that, GUG. GUG is the starting codon in prokaryotes like bacteria, blue-green algae, mitochondria, chloroplast, like that. GUG is the starting codon. The GUG name is formal methionine. Okay, formal methionine. During the time of protein synthesis, amino acid is recognized by more than one codon, the function is known as degeneracy. Degeneracy. Okay, every amino acid is coded with only three codons. Oh, three codons. I will explain. One amino acid, it is coded with 3 AUG, like this. 3 codons codes for amino acid. If one codon is coded with more than 3 amino acid, that type of phenomena is called degeneracy. Okay, that type of phenomenon is called degeneracy. The first recognized codon is UUU. Okay, this is the first recognized codon by Nirenberg. Okay, this is the main important starting codons of AUG, starting codons AUG and GUG. Synthesis of DNA. Synthesis of DNA from DNA. In my previous video, I explained this process of synthesis of DNA from DNA. Briefly, I explained this process is called replication or duplication or autocatalysis. It requires 7 hours. In this, DNA polymerase 1 catalyzed to the replication. If it is enzyme which is the first recognized by Kohlenberg, it is very very important, Kohlenberg. The Kohlenberg is known as Kornberg enzyme. In each replication, 2 daughter DNA are found. In each replication, one DNA to 2 daughter DNAs are found. Replication is known as, I already explained, autocatalysis. Okay, it takes place 7 hours. Semi-conservative method, it is explained by Watson and Francis Crick. Watson and Francis Crick. The newly synthesized DNA. The newly synthesized DNA. Okay, this is the newly synthesized, one is parent DNA. The newly synthesized DNA of replication is converted the half of the helical of mother DNA. During the time of this protein synthesis, DNA able to synthesize from mRNA. Okay, I will explain this as one chart, single chart. This is DNA. DNA gives another DNA. This is called bDNA. Okay, from bDNA, mRNA is synthesized. This is the table which I have made for easy to understand this. From this mRNA, another DNA will form. I will explain this only. This is called, already I explained, duplication. After duplication, the daughter DNA is called bDNA. Okay, bDNA. During the time of protein synthesis, DNA able to synthesize from bDNA, the mRNA is synthesized. This is known as transcription. Very important, transcription. mRNA formed from DNA, synthesized from DNA. This is called, the process is called transcription. Okay, this process is required 5 hours. This process required 7 hours. This process from mRNA to DNA synthesis takes place within 5 hours. The catalyzed by the enzyme is known as RNA polymerase. RNA polymerase. This is the catalytic enzyme. The each transcription of single mRNA is synthesized only from, previous video I explained you, there are the 2 helicals of DNA, 3' 5' 5' 3'. The mRNA is synthesized only from 5' 3' helical, 5' 3' helical. This is called synthesis of mRNA is known as template. This is known as template. The synthesis of DNA from mRNA is known as the synthesis of DNA from mRNA. Okay, this is called cDNA. Synthesis of DNA from mRNA is called reverse transcription. This is transcription, this is opposite, reverse transcription. This is not cDNA. This C stands for complementary. This C stands for complementary. This is abnormality. This is one of abnormality. It is no stabilized. No, it shows not stable character. It is recognized in cDNA means polygenic part. It is removed from DNA. DNA remaining immunogenic part convert. Okay, I will explain this. If cDNA is there, this is the DNA cDNA. This is called diseased DNA. This is a abnormal character. This is called diseased DNA. In the DNA, two parts is there. Okay, two parts is there. One part is called pathogenic part. Another part is called immunogenic. Pathogenic part, immunogenic part. Just imagine if disease is came when we remove the pathogenic part in normal DNA, nothing will happen. If we remove the pathogenic part, the immunogenic part synthesizes remaining pathogenic part and the normal process will going on. If in this condition, cDNA condition, if we remove the pathogenic part of the DNA, the immunogenic part is also converted into pathogenic part. Just like we observe in, this is observed in cancer cells. Okay, each cancer cell converts another cancer cell into another normal cell to the cancer cells. Just like this, cDNA, if we remove the pathogenic part, the immunogenic part also converted into pathogenic part. Okay, this is the dangerous gene. The disease caused part of DNA is DNA part converted into immunogenic, normal part is converted into the diseased pathogenic part. Another thing, it is we clearly observe in retrovirus, which is the main important cause of disease, AIDS. In the AIDS system, AIDS, the DNA is converted, pathogenic DNA is converted into immunogenic DNA. This is the this is called these type of DNA is observed in retrovirus families. Okay, this is the synthesis of DNA, RNA, tRNA, and retrovirus. The continuation of this lesson will be the next video. Before signing off, subscribe Think 360 for more updates. Like, comment on this video. Thanks for watching this video.